Hey everyone, in this video lecture, we will be talking about genetic mutations. So, throughout this unit, we've been talking about protein synthesis or the steps involved in making proteins. We know it's a two-step process involving transcription, where DNA is copied into RNA, and then translation, where RNA is copied into proteins. In this lesson, what we're going to look at is what happens if there's a mutation within the DNA. So, a mutation is a permanent change to your DNA sequence. And if the DNA has been changed, that then will affect how the protein is being formed or if it will be formed at all. And this is often linked to many genetic disorders and diseases. There are two kind of ways that people can get mutations. So one is natural causes, and this is where a person might inherit a mutation. So you yourself did not develop the mutation, it was passed down to you from parent to child. And the parent might have gotten it from, a natural cause themselves, or alternatively, we'll see in a minute, environmental based causes. However, it was passed down to you via your parents. Another natural way that mutations might arise is within the DNA gets copied, and your DNA gets copied before mitosis in order to make new cells that have the exact same DNA. Well, this process is really good, but there is a potential for mistakes to be made. And let's say it was supposed to copy the DNA and add a T, but accidentally an A got added. So that might happen where mutations could arise. Environmentally, meaning like external factors could cause mutations. So UV radiation is a big factor in causing mutations within the DNA. And this is because when you're out in the sun and your skin is hit with UV radiation, what that does is it damages your DNA. So in this picture here, it shows all the different UV rays hitting DNA, basically damaging those nucleotides. And what has to happen is when your DNA is damaged, the nucleotides are not functional. What will happen is your cells have a repair mechanism to remove damaged nucleotides and replace them with new nucleotides. However, this mechanism is not good at proofreading. So, what I mean by that is it often will make mistakes where the wrong nucleotides are added instead of the correct ones. This type of polymerase that will come through and add the complementary nucleotides is not very good. Similarly, with x-rays, the same thing can happen. Damaged nucleotides, we have to come in and fix them, as well as exposure to chemicals, things that we consume within our body, things that we put on our bodies. All of these things have the potential to cause mutations to the DNA. And there are many different types of mutations. Now, I do want to stress that mutations might happen to one single nucleotide within the DNA, or it could happen to 10, 20, 30, hundreds of nucleotides at a time. So pay attention to what we're going to talk about. It's the type of mutations. This could apply to one single letter, or it could apply to whole sections of nucleotides. So in this example, I have a sequence of DNA with no mutation. So the sequence should read GTAC, and then there would obviously be more letters either before or after this section. A substitution mutation is where one or more nucleotides are substituted for one another. So in this example, in the second spot here was a T nucleotide. Well, it was replaced with a C. It was substituted with a C. So substitution, just like a substitute teacher comes and works in place of your teacher, a substitution mutation is one nucleotide was replaced with another. An insertion mutation is when one or more nucleotides get added. So in this case, a G was inserted here. Now, what that did was it pushed the TAC all over one position. So the TAC is still there, but now they're in new positions within the DNA. And then the last type of mutation is a deletion, which is where one or more nucleotides get deleted. So in this case, the T from the original sequence was removed, and we still have the A and the C after where the T should be, but now they have been pushed back one spot. And we'll talk about the effects of these in a minute. So, substitution is a replace one or more letters or one or more nucleotides. Insertion is to insert or add one or more nucleotides. And a deletion is to remove one or more nucleotides. And so the effect of mutations is that, so in this example here, we have our DNA, the RNA, and the amino acid that should be added if this was a normal functioning protein. So no mutation occurs. The first effect of a mutation might be a silent effect, meaning that the protein is normal. It is functioning as it should. The change in the DNA had no effect on the protein whatsoever. So in this example, the DNA was supposed to be GTA, but it was mutated to GTG. So the G is a substitution mutation. The RNA now reads CAC. And if we were to look at our codon chart and see the codon CAC on there, it also codes for the amino acid histidine, which the codon CAU codes for. So in this case, the change in the DNA has no effect on the protein at all. So this is a case where the mutation has no effect. We call it silent because things kind of go on as normal. A different effect is called a nonsense effect. So in this case, the DNA was ATG. Now the DNA reads ATT. So still one single nucleotide was substituted. However, instead of the RNA reading UAC, it now reads UAA, which if we looked at our codon chart, UAA codes for a stop codon or stop. It's not an amino acid, it's just a stop. Whereas it should have been tyrosine as the amino acid. And then there'd be another codon after that, and so on. However, because of this mutation, we received a stop codon before it should be a stop codon. So what happens is with nonsense mutations, you end up with a shortened protein, meaning it doesn't have all the bits it needs to work properly. So it might have a little functionality, but it won't work as it should because it doesn't have the rest of the protein that it needs. And then the last type of mutation is called a missense mutation, where the protein is completely faulty. It doesn't work as it was intended to work. So in this case, the DNA was supposed to be GTA. Well, that G was substituted for a T. And so now the codon, instead of being CAU coding for histidine, it is now AAU coding for asparagine, which in this case, we are now adding the wrong amino acid. So this is where the protein is now not going to work correctly because the wrong amino acids are being added. So that makes it an entirely different protein. So this sort of effect of mutations tend to be where new traits might arise. So if we think ahead to natural selection, so within populations, right, if you have light colored fur on mice, all of a sudden a mutation like this appears where a whole new protein is created that might make a mouse have dark fur, and that might give them an advantage, and then they could pass that trait on and whatnot. So this is a case where fully new proteins might appear in a population, and those new proteins could have a benefit if it's a new trait, or what could happen is the protein doesn't work like it should, and that might lead to a disease or some sort of disorder. Now, that is just with one single amino acid changing. What you do not want to have happen is frame shift mutations. So, a frame shift mutation is where you get a nucleotide or multiple nucleotides getting inserted or deleted. And when that happens, as we saw earlier with insertion and deletions, it shifts the nucleotides over. And when that happens, that changes the way codons are read, meaning that not just one amino acid is wrong, many amino acids are going to be wrong. So this could be very detrimental to the functioning of a protein. So in this case, we have a normal amino acid sequence. We have its normal protein here. Well, let's say one single letter gets deleted. So where this codon was UUU with phenylalanine, one U gets deleted. That pushes every nucleotide over one spot to the left. So instead of UUU, we now have UUG, which is the amino acid leucine. The next codon is GCA, which is the amino acid alanine. Then we have UAG and a stop. So the rest of this protein is completely incorrect. Only the first two amino acids are right. And if we think about this and compare this to like words in a book, words in a sentence, let's say we delete one single letter in this sentence. So we have the sentence, the fat cat sat. All three words, or all the words are 3 letters long. Well, if we deleted the T and moved every letter over one spot, keeping 3 letters per word, this now becomes gibberish. And it's the same sort of thing with frame shift mutations. Deleting or inserting one or more nucleotides changes the entire way the ribosome is going to be reading these codons, which changes the amino acids entirely. So this can be very detrimental within a cell if you're trying to ensure that you have functioning proteins.